Alrighty, so our group had Klinefelter syndrome, and so our first question is, what simple genetic test is used to define or diagnose someone with Klinefelter syndrome? So in our case study, we had a 15-year-old male, and he came to the doctor because he was concerned about his female characteristics. He ended up having small breasts and round hips, and so the doctor ran tests, even though he was pretty positive about what it already was. But he still ran tests, and the results were Klinefelter syndrome. And so this disease is just caused from non-disjunction, and the male receives an extra X chromosome, so his genotype would just look like XXY. So how do we diagnose somebody with Klinefelter syndrome? So we use a karyotype test, and so a karyotype is just an individual's collection of chromosomes. So it's literally just a test on your chromosomes, or of your chromosomes. So in order to do this, there's 5 steps. You start with a sample collection. It can be blood, tissue, amniotic fluid, or any other bodily fluids. Usually blood or tissue is the 2 most common. And then second step is arresting cells at metaphase by a drug called colchicine. And so the tissue or the samples are sent to the lab, and the cells are arrested by this drug. So are basically just stopped by this drug. And then third step is swelling, separating, and spreading of chromosomes, which is done through the use of a hypotonic solution, which we can see here. The cells are centrifuged and concentrated into layers. And then the fourth step we have is separating these the chromosomes onto a slide, which can be used to put under a microscope. And the fifth and final step is staining the chromosomes, which is used by a process called Geisma, and it's just an easy method, a quick and easy method to stain chromosomes, because we need a quick method since a karyotype test is done all by hand. And so this is done underneath a microscope where a picture and an examination of the karyotype is taken.
Good morning. So our group had Klinefelter syndrome, and I started at question 2, which asks how chromosomes determine sex in humans. The sex chromosomes, or pair 23, are the chromosomes that determine the sex of humans. Females have 2 X chromosomes, one from each parent. Males, however, receive an X chromosome from their mother and a Y chromosome from their father. Unlike the other chromosomes found in cells, the Y and X chromosomes control only sex-determinate genes. I also had question 3, which asks if people with Klinefelters appear more male or female. Technically, those with Klinefelter syndrome are male, but because they have an extra X chromosome, they exhibit traits that are typical to females as well. For example, they may develop breasts, a shorter torso, and broader hips. Puberty can also be delayed or incomplete. Infants with Klinefelters appear male, with some differences in motor development and speaking. Once they reach the teenage years, it is more apparent that something is different. They are later to puberty and often do not develop fully in areas such as facial hair and muscle tone. In the adult years, however, they appear much like normal men, except they are often taller than average height.
Alright, so briefly describe how non-disjunction during meiosis leads to this condition. So, non-disjunction is an error in the process of meiosis. Looking at the little diagram on screen, normal meiosis happens when an egg cell is multiplying, basically, and its daughter cells in the end will have 1 X chromosome. But unfortunately, non-disjunction happens when the chromosomes don't split correctly, and instead the resulting daughter cells will have an extra X chromosome, leading these 2 daughter cells with 2 X's and these 2 cells with no X chromosome. If these 2 daughter cells over here are fertilized, this will result in a male having Klinefelter syndrome. In our case, there are a couple other options that could happen, but for the purposes of our topic on Klinefelter syndrome, we're going to assume that the person would end up having Klinefelter syndrome. Little fun fact though, if one of these two daughter eggs are fertilized having no chromosome, or no X chromosome, this would result in something called Turner syndrome, which is different, but just a fun fact. Is Klinefelter syndrome an inherited disease? And if not, can it be considered a genetic disease? Explain. So, Klinefelter syndrome is not an inherited disease because non-disjunction happens at random. There is no way to predict if a mother's mitosis will happen perfectly or normally. It honestly is just a card draw of luck to determine whether or not somebody is going to have Klinefelter syndrome. But is it a genetic disease? Yes. A genetic disease, by definition, is an illness that occurs when a change in DNA happens. An extra X chromosome, compared to the usual 2, is a difference in DNA. So, by definition, Klinefelter syndrome is a genetic disorder or disease. Finally, question 6. In this case, would one of the X chromosomes be inactivated and form a Barr body? A Barr body basically inactivates one of the 2 X chromosomes, and the reason a Barr body happens is it stops the Barr bodies regulate the amount of X-linked gene product being transcribed, so basically it might stop the amount of estrogen being produced just to ensure there's a healthy balance in somebody. But even though a person with Klinefelter syndrome might have a Barr body in one of their X chromosomes, this will not stop the disease entirely. The person still will have Klinefelter syndrome's symptoms, but this will happen no matter what. In fact, all females on the planet actually have a Barr body in one of their X chromosomes already. So it's completely normal for ladies, but unfortunately for those with Klinefelter syndrome, a Barr body will not stop their disease even if it inactivates an X chromosome.
Alright, now to summarize our presentation, we have sex chromosomes, non-disjunction, Barr bodies, and then the karyotype test. So we all know that females and males, in a female we have XX chromosomes, and then in the male we have XY chromosomes. And then in Klinefelter syndrome we see that the male has an extra X chromosome and his genotype is XXY. And this is just caused by non-disjunction, which is the failure of chromosomes to split properly during meiosis. So this also backs up that the disease is genetic and not inherited. And so as far as Barr bodies, one of the X chromosomes will just naturally inactivate as it does in a female. But for someone who has Klinefelter syndrome, the more feminine features will be present if one of the X chromosomes does not inactivate and cause a Barr body. And lastly, karyotyping is how we will determine if somebody has this disease. And it's just a simple test that is run in a lab. And that is the overall overview of Klinefelter syndrome.